So we're going to look at the cell model here for Bio 105. So going through the parts here on your laboratory manual here, it starts off by saying that you have to know each of the structures and then also know what their functions are, and that's what I'm going to go through here. So these are the ribosomes. They're shown here and here. So these little red dots. What ribosomes do is they synthesize or they make proteins. All right. Next is the nucleus. So this whole structure here is the nucleus. Our nucleus houses our genetic material, and it's basically the control center of the cell. Within the nucleus are these little hair-like structures, and that's chromatin, and that is our normal form of DNA. All right, so the DNA when the cell is not dividing. When the cell is getting ready to divide, it will package up that chromatin into something you're more familiar with, and those are chromosomes. Next is the rough endoplasmic reticulum. We can see rough endoplasmic reticulum here and here and here. The reason why we call it rough is because it's studded with ribosomes. All right, and what that rough endoplasmic reticulum does is it packages the proteins that are made by the ribosomes into small membranous sacs called vesicles. And these are trying to show some of these vesicles over here. All right. Next is a smooth endoplasmic reticulum. This is smooth endoplasmic reticulum here and here and here. So it looks similar to the rough endoplasmic reticulum, but it lacks the ribosomes on it. And what the smooth endoplasmic reticulum does is that it makes lipids. So lipids, fats and oils, phospholipids, so those sorts of things. Next is the Golgi Apparatus. This is the Golgi Apparatus. Now what the Golgi Apparatus does is it modifies proteins from the rough endoplasmic reticulum. So ribosomes are going to make proteins. We're going to package those proteins into vesicles. Those vesicles are going to move to the Golgi Apparatus, and they're going to be modified some way. So maybe a sugar group is added to it, or a lipid group. All right. So after that happens, if it's something that the cell wants to get out of the cell, so such as tears or hormones, what will happen here is we'll take that product and it will fuse with the cell membrane. The cell membrane, the vesicle will open up and it'll release those contents. And so that's what this is. It's a secretory vesicle. It's secreting substances out of the cell. Next are lysosomes. Lysosomes are these kind of purplish round structures. Lysosomes contain digestive enzymes, and those digestive enzymes can serve as a defensive function, or they can be used to break down old organelles. Next are fat vacuoles. So these are fat vacuoles, and a vacuole is just a large vesicle, so a large membranous sac, and these guys contain, well, fat. Next are centrioles. These guys are centrioles right here. They kind of look like licorice. So what the centrioles do is they guide the movement of the chromosomes during mitosis. And then lastly here are the mitochondria. Mitochondria here, as I sometimes tell my students, look like circus peanuts, they're a little candy. But what they do is they produce energy for the cell, so making ATP.